So last time we went over the current source density, so the derivation of the current source density. So remember that's basically just a single number at each point. So, and what it represents is there current sort of erupting out of a spot, or is it sort of disappearing into a spot at each point, or is it just going around in a circle, maybe not going in or out? So it's just a single signed number that just tells you whether there's current sort of coming out of a spot or going into it and disappearing inside the cell where you can't record it. And so that was the current source density, and you calculate it by doing this thing called Laplacian, which is a second spatial derivative of this field of potential / voltages. So that's all kind of abstract, and so what we want to talk about today is current source density data. So what is the actual data that people have recorded and done current source density on that suggests that we should model activity in the cortex as a radial dipole. So a couple lectures ago I said, how do you actually sort of see any kind of evoked potentials at the scalp? What do you need in order to get some current to come through the scalp into the electrode? And the argument was that we needed to have these radially, elongated neurons that are all lined up with each other. So they have to be elongated and they have to be lined up with each other, and then they have to be stimulated in a laminar fashion, like maybe just at the bottom of all the little vertical cables or at the top. And we just said that, but I didn't give you any sort of reason like why, what's the actual data that suggests that that's actually the way it is. And what that data is the current source density data. So we'll talk today about the current source density data that suggests that it is a good thing to actually model electrical activity that you can record at a distance as a radial dipole. Radial meaning like perpendicular to the cortex. So what are we talking about? So we're just talking about we're zooming, pretty far in. So if here you have some cortex like that, I drew it a little bigger than it is, so you can see what's going on. What we're talking about now is just a little chunk of that cortex. So if we just take a little chunk of it, like a couple millimeters of it, and so take that little cube out, and then we'll draw it here for doing some recordings from it. And what are the kinds of recordings that we're doing? We're typically using a microelectrode for this, because this is current source. I'll first talk about current source density actually in the cortex. And I mentioned last time that an electrode, a microelectrode, which is a very fine insulated wire with a conductive tip, doesn't really do much damage to the cortex, because the cortex is kind of like felt. It's kind of like all these little wires that are all sort of, even surprisingly, even if you might think, well, what about the blood vessels in there? Wouldn't you pop the blood vessels if you stick an electrode down in? And actually, if you come in with a fine electrode like this and aim right for a blood vessel, what typically happens is it'll just push the blood vessel off to the side. It won't actually pop it. And so it's actually, you do surprisingly little damage to the cortex if you put a fine wire into it. If you want to find out where you were recording, it turns out you have to actually make a little micro lesion by running some current through the electrode. Otherwise, if you just pull the electrode out and then kill the animal, section up the brain, there'll be no record of where your recording was. Now, that's a microelectrode, so microelectrode. In humans, they often use much bigger electrodes. And the reason for that is if they're trying to get to a certain place in the brain, they need an electrode that's more rigid. And so those, by the standards of animal recording, are kind of like telephone poles. Those guys actually do damage because there might be like over a millimeter in diameter. And so if you put one of those down into the brain, it's definitely going to do some damage on the way in. But the microelectrodes actually don't do any substantial damage. So when you hook up the signal reference to ground, and what is ground? Usually ground is like some, another electrode that's on, on the skull or on the skin someplace. You get recordings of potential or voltages. And so what that looks like is, there'll be a spike in it occasionally. So there's a little spike that occurs, and then you come maybe another spike, and then there'll be a change in potential and maybe another spike. So the spikes. So this is this is the potential, going up that way, over time. So the spikes are upside down because that's the electrode is outside of the cells, and so when the sodium rushes into the cell to start a spike, some positive disappears into the cell, and that appears as a negative potential. So typically what you do is if you want to see the spikes, then you do a high pass filter, which means filter out the base. Just leave the high frequencies. And in that case, what you'll get is you'll just see the spikes. So you'll filter out that low frequency change. So you'll just see, just the spikes. And so and then usually you flip them around so that they look like regular spikes. So that's if you're doing single unit recording, trying to sort of isolate individual cells. But alternatively, you could just do a low pass filter, which means turn the bass up or, turn the treble down. So if you do a low pass filter on the same exact signal, then what will happen is you'll get rid of the spikes because the spikes are high temporal frequencies. So we're doing a temporal filter. And so in that case, you'll just see this local field potential. So this is usually called local field potential, LFP, local. And that's the basic data that we're going to be talking about using. So local field potential, and here's spikes. So they're both in the same. The spikes, of course, will come and go depending as you move the electrode because you typically move the electrode in tiny little steps until you get close to a cell, and then then the spike can get quite big if you're right up next to a cell. But you don't want that. You don't want that spike signal because what you really want is kind of like the summed. This is like summed extracellular currents. So you want the roar of the crowd, extracellular currents. So these are all currents caused from all these different cells simultaneously becoming active, and you don't really want to just pick out one and have that contaminate the data. So this is the kind of data that we're typically recording in order to do a current source density calculation, the local field potential. And like I said, it's there anytime you put an electrode, anywhere in the brain, you're gonna see that. So then let's just go to a little chunk of the brain, and we'll start off with a 1D current source density. So current source density, the way you estimate it is with the Laplacian, which is a second spatial derivative. But don't confuse that. I said it last time too. Don't confuse that with the dimension that you're doing this Laplacian in. So you can do a second spatial derivative of a one-dimensional set of data, or a two-dimensional set of data, or a three-dimensional set of data. So we'll just start off talking about a one-dimensional current source density calculation. And so what is it, what does our starting point look like? I mean, what does our recording site look like? So here's a little chunk. So this is this guy blown up there. And so we would typically do this by, putting an electrode. So, here's an electrode. So putting this electrode down into the microelectrode down into the cortex, and then you typically would put it in and then pull it back out and record the response to an event. So basically you would sort of put it all the way down to the bottom and then sort of slowly pull it back out. And so what are we recording? We're recording in different layers in the cortex. And I gave a little diagram of some of the layers in the cortex. So you have, let's make that electrode go in right about there. So, you have the input layer, and then you have the various layers outside of the input layer where the information gets spread out to after first coming into the input layer. And so the input layer is layer 4, and then you've got these other other layers. So here's layer 1, layer 2, 3, which layer 4, that's the input layer. 5, that's a motor output layer. 6 is one of the feedback layers. And so, what's in there? Well, you've got, these pyramidal cells, a bunch of pyramidal cells that are, in there spreading out their dendrites. What is the basic circuitry that we talked about? So we said some input comes in and it goes to layer 4. That's the input layer. And then from layer 4, the cells kind of project, out to, other layers. And some of those other layers, like layer 5, come back out. Some of the layers, like layer 2, 3, go up to higher areas. So this is, motor. This is, higher. This is input coming in. And some of the layers go, like layer 6 goes back to feedback. And so when we put the electrode in there, we're going through all these different functional layers in the cortex. And what we're typically doing is doing some kind of event-related recording. So we're recording, so we do some very short event. It could be like a click if you're in auditory cortex, or a little flash if you're in visual cortex. And then we do that like 20 times, say, and then average it all in order to clean up the signal. Because, maybe the animal wasn't paying attention that particular time you flashed, and so you won't get much of a response. So we record the voltage trace out of that electrode for each layer. And so for each recording spot, we've got a recording over time. And so I'll draw what the data looks like over here. So we got, so here's a recording of depth versus time. So this is time going in this direction, and then depth. And so we're doing a one-dimensional current source density with depth. So here's when we actually started the stimulus. So this is the event right there. And so what does our data look like? Well, it just looks like a bunch of traces. So, and we're averaging 20 times to get some of the noise out, but basically what you'll see something like, there'll be some, kind of signal like that over time. So what are we, what are we recording? It's the potential. So this is, the potential, but what is the axis? So this axis is time. So that potential is just right here. Okay, so it's just it's just how high up and down that little blue line goes, but this direction is depth. So that's what we're doing our current source density over. So depth by time, and then at each depth time point we have this. At each depth we've got this recording of what the potential was, positive or negative, because it can be positive or negative. And so if you go to a different depth, you'll get a slightly different signal. So say here's a slightly different depth, and it turns out you can see that this evoked potential changes quite rapidly. Even a small advance or retraction of the electrode, like 1/10 of a millimeter or a 1/3 of a millimeter, the evoked potential will look completely different. So you'll have a whole bunch of these records over time for all these different depths. So this is our raw data, which is the potentials over time. So, and what does that represent? It just represents the summed currents flowing all around in the cortex from whatever happened in the cortex. So it's all the extracellular currents caused by all the activity of nearby neurons in the cortex. So now we need to calculate the 1D current source density. And the 1D current source density calculation is, we calculate the Laplacian. And what the Laplacian was the second spatial derivative. So we do that operation on the potential. So we start off with potential, and then what we end up with is, we end up with a number which is the current source density, and so that's the Laplacian of the potential at that depth. So we can do that calculation, for each depth. So each depth there's going to be a graph. So let's just do. And then we said, so what is the Laplacian? The Laplacian was the second spatial derivative of something, the potential. And what is the second spatial derivative? It's basically the curvature. And how do you estimate the curvature? Because the first derivative is the slope, second derivative is the curvature. How do you estimate the curvature? One easy way of estimating is just take the average of the neighbors in space. And space in this case is just depth. So this is, so depth is space. So here's depth. Yeah, space 1D. So you just take the average of the neighbors and subtract it off of the current point. So let's just do it for like a single point, like right here, for this guy. So here's the point that we're calculating the current source density for. And then what do? We take the average of the spatial neighbors, and so that would be this one, and that would be that guy, and that guy. So and if we average those two, you can see they're a little bigger than the central point. And so then if we subtract off the average of these two guys from the center, we get a negative number. So if here's the zero of our current source density, we're going to end up with a negative number for that point. So right there. So we just did a single computation for that point in depth, and that's what we got. We got a current source density, a negative current source density. So here's the first thing I mentioned last time. All of those were positive potentials, and so you can't you have to do this calculation to sort of like actually figure out, is their current sort of disappearing? So even though all three of those points were positive, when you do the current source density calculation, you get a negative current source density, which means current is disappearing into that point, even though the voltage at all those points is positive. And so if you do the current source density calculation, then you'll just end up with another whole set of current source density, numbers like this. And they'll look different than the voltages. And in fact, they'll change even more rapidly with layers than the voltages do. I'm not drawing it accurately, but if you look at some of the papers and the readings, you can sort of see what this looks like. So you'll have a whole... And what these represent is basically, at each point in time, so it's still time here, and it's still depth. So at each point in time, for every depth, what is the current source density, meaning is current sort of disappearing in that part of the cortex, or just erupting out of the void of the inside of cells. So this is here's our Laplacian second spatial derivative of the potential, and here's the potential. So the first thing you can see when you do this was done like 30 years ago, 40 years ago. The first thing you can see is that the current source density changes very rapidly with depth. So you might go down like 1/3 of a millimeter, and suddenly the current source density is positive. There's current erupting out of that part versus disappearing into that part. And so that suggests that there's a lot going on with depth. But what about if you go to some other part of the cortex? So if you go to some other part of the cortex, so there's here's the two other dimensions of the cortex. You've got so we have the depth of the cortex, but what about just doing the same thing at a different place? So we did it over here. What if we just put the electrode over here and try the exact same thing here, and just find out what's going on in that part of the cortex? What happens then? And what's generally changing when you go sort of across the cortex, tangential to the cortex, parallel to the cortex, instead of going through the cortex? Generally, if you're in an early sensory area, there'll be something like a visual map. So for example, a visual map. So it might map different parts of the retina. So if you take the electrode out of there, go to some other part of the visual map, make sure that you're stimulating the part of the retina so that you get a response. Obviously, if you don't stimulate the right part of the retina, you won't get a response. But if you just make a big flash that covers the whole retina, and then do the exact same thing again, what you'll generally find is it looks pretty similar. There'll be the same kind of current source density changes with depth. But the overall pattern of current source density at different depths is not that different. And that fact is the fundamental reason why we think that it's good to model what's going on in the cortex electrically, or at least the part that we can actually detect from the scalp, as radial current flow. So either current flow kind of up or down in the cortex. And the fact that the current source density doesn't change very rapidly, the pattern of current source density with depth doesn't change very rapidly when you move to some other part of the cortex, suggests that we don't see those sudden changes as you move, to different parts of the nearby parts of the cortex, suggesting that there aren't large, like lateral flows of current that are detectable at a distance. So this is the fundamental observations, actual data, that suggests that thing I said before, that, how do you see, current at the scalp? It's because you're getting this coherent activation of these radially aligned pyramidal cells that are perpendicular to the cortex. This is the data that actually really supports that idea. Okay, so this is the event here. Yeah, event. Okay, so any questions about so 1D current source density? So typically, if you do, I often just illustrate it, just subtract the average of the neighbors. Typically, what you would do you would get a nice smooth curve, like fit a spline to the data through depth, and then you could analytically take the Laplacian of that spline and get a nice smooth current source density curve. But I think it's important just to keep the basic computation in mind. And the basic computation in mind is the second derivative is just subtract off the average of the neighbors from each point. And you would just do this. You would do the same thing. To get that single curve, you'd have to do the same thing for each one of these points on the potential curve. Okay. So all good about sort of this. Now, we haven't said like how it relates to cells. And so what I want to do down here is kind of like make a diagram of how how we actually see how we think it actually relates to the actual pyramidal cells that are in the cortex. And so I'll draw out first draw out a chunk of the cortex here, make it a little bigger than last year so you can see it a little better. And so here's where our event is occurring. So there's the event, and then I'll shade in the sinks. I'll shade in the sinks and leave the sources as an opening. And so sources and sinks, that's just positive and negative current source density. So if we do this kind of calculation here, and then kind of color it in basically by, whether it's positive or negative, that will tell us kind of where sources and sinks are. So what is the actual data look like? This data look like? So this is basically, let's just colorize that data. And so what it looks like is we got something like that, and something like this, and then and then a smaller one over here. And so I'm going to color in. So we'll color in the sinks. So this guy's a sink. And a sink means like a negative current source density where current is disappearing. And this guy's a source, not colored in. So then there's another source down here, another sink down there, another sink up here. So this is the same thing. This is over time, and this is depth. This is layer 4. layer 2, 3, and then layer 1 there, layer 5 and 6. So that's kind of the pattern that you see across many different cortical areas. This has mainly been done in sensory cortical areas where you can do a nice event, a nice sharp event. But what do we think is actually causing that, this pattern of sources and sinks? And so let's look at and generally, where, what direction does the current flow? It flows from the, as you might expect, the source to the sink. So let's take this first one here. So the first one, the strong one that you can see, is basically a source right here going. Let me just make a little hole for myself. So that's a source going to a sink. Okay, it's not contrasty enough. Let's make that one. So there's a source going to a sink. And this is the fact that that's the first one you can see is a little disturbing, because it turns out if you go into layer 4, a lot of the cells in layer 4 that are hit by the input, say if for example, dorsal lateral geniculate nucleus, that's a visual nucleus that goes up to layer 4. A lot of those cells in layer 4 are stellate cells. They're not elongated. And so they're the kind of cells that, unfortunately, we can't easily see a signal from because there isn't a separation of the source and the sink. Some channels get opened up all over the stellate cells, and then current flows to the center of the stellate cells, but it's all sort of symmetrical, and it kind of cancels itself out. It's not like an elongated, like we stimulate the basal dendrites and the current flows up. So unfortunately, the very first stage of where there's really active geniculate cells synapse on to the stellate cells in layer 4. I can't see that very well. Sorry about that. But then after that, very soon after that, we see this big sort of source in the upper layers going down to layer 4. And what is that being caused by? And so that's being caused by basically if we have some, pyramidal cells that are, say, in layer 4 or above layer 4, and the part that we can't see is, something came in and synapsed on one of those stellate cells, and then the stellate cells synapse onto these guys. Can't see that part, but we can see the part where the pyramidal cells got activated. And what's the part we can see? Well, we can see some current sort of disappearing in here, and then coming out up here. So this guy is the source. This guy is the source, and this guy is the sink. So it looks like current is flowing, from the source to the sink. That's that first strong kind of current dipole. And this is what we call a current dipole. So we're modeling kind of like what's going on in the cortex as this radial current flow, and this is the radial current flow. So it's basically the stimulation of the basal dendrites of these cells, and because they have a very good membrane that insulates, the current will flow up, and then it'll take a while before it sort of flows out, and then we'll see a source of positive current up here, and that would that would correspond to generally what somebody in the evoked potential literature would call a P1, positive 1. And why is it positive? Well, if here's here's the, the skull over the top, and there's an electrode recording it. What you're going to see, you're just going to see, just some little, positive current appearing there at that electrode, assuming that the cortex was actually parallel to the skull. Of course, it isn't all the time. A lot of times the cortex is perpendicular to the skull, in which case, it's going to cause something else. But on average, we see a P1 because even though we can't see this initial layer 4 input, that's the first sort of very visible input with these elongated pyramidal cells, and we see current sort of going up in the cortex, positive current, and erupting out of the top of the cell, and that makes a positive signal. So there's the P1, and so then what happens? So then what happens is we get an N1, and so the N1 is going from this source to that sink. So this is another source down there. And so what do we think the N1 is? Well, the N1 is basically a sink on the apical dendrites of these very large cells that are above and below layer 4. So especially like some of these deeper layer motor output cells in the cortex that go down to the striatum or the superior colliculus or the spinal cord. Those guys are big, beefy pyramidal cells down in the lower layers, and those guys are now have the opposite sign of source and sink. So what's happening in these guys is you see a sink up here. So that's a sink, and then you see the source down here. And so what is that from? That's from this additional circuitry. There's a lot of circuitry. Not only will layer 2-3 go out, but layer 2-3 will go down and synapse on cells in layer 5. So that we can, that's generally the scheme of something comes into layer 4 and then it spreads out of layer 4 above and below, and then the above and below guys communicate with each other. And the bottom line is that many cortical areas behave the same way. So you see this initial sort of positive signal from the activation of cells that are a little bit more up in the cortex, a little more shallow in the cortex, and then later on you see these bigger bruisers, and so this would be, what an evoked potential person would call an N1, and so we've got a sink here, so that's things disappearing because it's a negative current source density, and a source down here. And you can think of the current as basically flowing, from the source to the sink, like that. It's easy to get confused about the sign of things. So this is P because the source is going to be positive current erupting from the top. But when we think about the direction of the current flow, it's actually from extracellular current flow. So this is, extracellular, which is what we're recording. So the extracellular current flow is going that way. This return loop, but that appears as a positive number at our electrode. Yeah, so many sign changes. Okay, so this is the N1, and then if we continue a little bit later, you'll see another reversal in all cases. This is layer 4 here. So at a little later in time, you'll see some additional sources and sinks that reverse in sign again. So, and what those look like is some sinks down here, and sources coming out, and they slightly cancel each other out because this this sink right here will slightly cancel out this source over here. And here's a sink. But bottom line is that we see another positive wave, and the evoked potential person would call that like a P2. So basically this describes kind of how the cortex in general works. So stuff comes into layer 4, you can't really see that. Then it kind of starts to activate these more superficial pyramidal cells. That gives you a positive wave, and then the deeper, larger ones get activated. That gives you a negative wave, and then deeper and superficial get activated in yet the opposite way. And so you end up with kind of a classic, evoked potential picture of we've got a signal about 0, and then we start with a positive, and then you end up with a negative one, and then a positive one might even go back and forth again. And so that's your typical evoked potential signal from a cortical area. And it's all based on a 1D current source density, 1D Laplacian, 1D second spatial derivative. So what about what about another cortical area? Well, if you go to another cortical area, what are you going to see? Well, you're going to see something that's slightly offset, right? Because maybe the signal took a little longer to get there. So you'll have this might be another cortical area, like that. And so what are you going to see when you record? So this is, different cortical areas. So what are you going to see when you record at a single scalp electrode? You can see what the problem is now. You're going to add all these up, and then you're going to get our, evoked potential, ERP at, one electrode. And what's that going to look like? Well, sort of the same thing, right? It's going to look like, we wait, and then there's, maybe something that kind of looks like that. But it's going to be hard because these are all offset in time to figure out what's actually going on. That's really the reason that we want to do some kind of localization, which we'll talk about on Friday. So I'll talk about the forward solution and the inverse solution on the 8 o'clock and the 9 o'clock. So you might want to come to both of those if you're interested in the localization things. Okay, so that's 1D current source density, but what about 2D current source density? So we could do 2D current source density. And the way we would do that is rather than putting an electrode in, so you could try to put an electrode, a whole series of electrodes into one layer. But that's going to be quite hard to, because the current source density changes so rapidly with depth, that's going to be extremely hard to do, to find the exact same layer, because even if you're off by like a third of a millimeter or a tenth of a millimeter, you're going to get a different signal. And so typically what people do instead is they just put an array of electrodes on top of the cortex. So you could put a flexible array of electrodes that just touches the top layer of the cortex and then try to record evoked potentials, and you can do that. And sometimes in humans, they put these arrays in of much bigger electrodes on top of the cortex. But in an animal, you could put a fine array of a fine mesh of electrodes. Now, I said when we go through layers, we see big changes in the current source density, and when we go parallel to the layers, we don't see that big changes. But there are changes because, of course, we have different cortical areas. And that's obviously the signal in V1 and V2 is not going to be the same. Maybe it'll change the same with depth, but if V1 projects to V2, at least we're going to expect the signal in V1 to be delayed a little bit, V2 to be delayed a little bit relative to V1. And so for that purpose, we can do a 2D current source density. And so what that is calculate the second derivative with regard to space that's parallel to the cortex. And so if you just imagine like, I'll draw 4 heads here. And so first we have our data. So here's our evoked potential data. And so now we're looking down on somebody's head. Say this is their nose there. So you're looking down on somebody's head, and you record a whole bunch of different electrodes, on the head. So here's a whole series of electrodes that you've put on the head. And the current source density calculation is, remember, it's just something that's instantaneous in time, right? So you're just recording for one time point. So this is time point 1. This is time point 2. Time point 1. Time point 2. So here's our potential, and then here's our Laplacian second spatial derivative of the potential. And so how do we do that in 2D? We basically just subtract, each electrode from its two-dimensional neighbors. So it's the same. It's the same mathematical. So sorry, we subtract the average of the two-dimensional neighbors from the center point. So how do we calculate this guy? We just take, like, let's go out in the x and y direction and average those guys. Go out in the, you go out in the x direction, plus and minus, average those guys. Go out in the y direction, plus and minus. Average all the neighbors, and then subtract that from the center point. That's essentially what this operation, this Laplacian operation is. And so after we've done that, we then end up with a single value for that point at time t1, and then just do the same thing at time t2. So what would you end up with in this case? You'd end up with a spatial picture now, a two-dimensional spatial picture of sources and sinks. So over time, again, it's going to be a more complicated picture than your basic evoked potential picture, because as we mentioned before, if you have a positive evoked potential, that doesn't necessarily tell you whether it's a source or a sink. So you could have a situation, if here's 0, you could have a positive evoked potential, but that might have negative curvature indicating it's a sink. Or you could have a positive evoked potential with positive curvature indicating it's a source, and vice versa. You could have sort of the same thing down here. So you're generally going to get a more complicated picture. So this, that indicates, sink, even though it's positive. That indicates a source. This indicates a sink, and that indicates a source. So here's, positive and negative, potential. And so generally you're going to get sort of a, now, what is it telling you? So it's not really a source localization technique. So if we look at, let's just take an example of, so here's, draw it big enough this time. It's a nice big brain. Okay, so say we recorded an evoked potential that looked like something like this. Okay, so that's an instantaneous, instantaneous evoked potential. So here that's, at one time point. So all of these things are at one time point. So, what could have caused that? So if we do our current source density calculations, if we do them over here, you can see what's going on here. That guy is a sink because the neighbor average is bigger than the center point. So that indicates. So this guy indicates a sink over here. So current was disappearing. What about this one over here? This guy sort of indicates if you compare to these neighbor points over here, this guy indicates a source. And so you can sort of think of a two-dimensional current source density as, let's just say the cortex was parallel to the skull. It isn't, but let's just say it was. If we do a 2D current source density, it will tell us essentially where current is coming out of the skull and going into the skull. Now, some animals have a very flat cortex, in which case, if you did a current source density on top of their scalp, you would see something that approximates where things are coming and going, from in the cortex. In a human with a folded cortex, it's not as easy. So there's several things that could cause something like this. So you could have something that, like, just take some examples of what might cause this. One thing that might cause this is, say we've got a cell that's kind of like that. So it could be that what's actually going on is that this cell was activated in its basal dendrites, and it's actually causing this signal, like current is disappearing here as it goes into the basal dendrites, and then it's actually sort of appearing over here as it comes out of the apical dendrites. Maybe the cortex was just folded, that you're in a sulcus here. But it could also be something else. So it could also just be that you had a cell over here. I'll dot this one. So you might have had a cell over here and another cell over here. And it could be that, some current at this part of the scalp, was disappearing into that apical dendrite because maybe the apical dendrite was stimulated, but this cell, which is in a different cortical area, actually had its basal dendrites stimulated, and so this guy is going to make a positive signal. So you can't really distinguish, like, was it a sideways cell that got activated at its basal dendrites, or was it a sideways cell pointing the opposite way that was, activated at its basal, that was activated at its apical dendrites, or was it two cells, one of which was activated at the basal and one of which was activated at the apical dendrite? That's the reason why you want to do actual source localization when you have a folded cortex. But even that aside, you can see that doing the current source density by calculating this curvature, which is relatively hard to eyeball off of the evoked potentials, it's going to give you more information in general about where the current is flowing. It's not a localization method, though, when you have a folded cortex. Works pretty good if you've got a flat cortex. Okay, so that's 2D current source density. And then, like I mentioned last time, what's the gold standard? Well, you could actually use current source density for localization if you could do 3D current source density. So all you have to do then is, and I've got a paper in the readings that describes doing this in the auditory system of an animal. Well, you can see what the problem is. The problem is, well, you just need so much data. So what you need to do is you need to go into a cube of brain tissue and then, you know, put an electrode in over here and then record at all those points, and then put an electrode in over here and then record at all these points, and then put an electrode in, you know, over here and record at all these points. And so you would need like a crap load of data to sort of like completely fill up a cube of data. And at each point, you would do like 20 repetitions of the stimulus and average it in order to actually get your potential. And then you could do a 3D current source density. And what would that be? That would just be sort of go to a point and then see how the potential changes in X, Y, and Z. And then take the average of the neighbors and subtract that off the center point. And so if you did that, then that is a localization method. You would actually see where all the currents went. You know, did they, you know, sort of come from this nucleus and they went into here, and then they sort of erupted out of this nucleus. You could actually sort of make a movie of exactly where the currents went. The only problem is, like I said, it's kind of hard to do in practice. People have, you know, thought up all different kinds of tricky ways. Is there some way we could make very fine but strong electrodes? The problem is, like, if you make a very fine electrode, it's not very strong. And so it's hard to get into the cortex. There's something called the Utah electrode, which has been around for like about 25 years. And what they did was they made a little kind of pin cushion of electrodes. It's just got like 100 electrodes coming out of this base, like that. So it's basically sort of kind of like a little pin, like a 10 by 10 pin cushion of electrodes. Now, if you take that array they're very sharp and they're very, very small. If you take that array and try to push it down into the brain, it won't go. What will happen? It'll just push the brain down, and all bad things will happen. So what they figured out was, if I mount this electrode in this air pressure powered gun that moves it really quickly, there'll be enough inertia of the brain that it can sort of penetrate into the brain without damaging the brain. And so you basically have this kind of like air powered thing. You just go boom, and just like pop it in really quickly. And they've even used this in some cases for people, like say somebody's blind and they have no more visual inputs. You could pop an electrode like this into the visual cortex in order to stimulate different parts of the visual cortex. The only problem with this is that the recording site is just at the very tip of each one of those electrodes. And so that would only give you really a 2D current source density. It wouldn't give you a 3D current source density. What's the problem? Well, if you make a bigger electrode that actually has multiple contacts on it, people have done this as well. So if you make an electrode with multiple contacts, the problem is you need wires for each of those contacts. And so, you know, each of those contacts needs a separate wire. And so before long, the electrode sort of is no longer micro, it starts to get bigger. But you probably know Elon Musk is trying to sort of make electrodes like this that have lots of contacts, so that you could actually, you know, stimulate the brain or record from the brain in more detail. Nobody's really made something very practical yet that can sort of record in 3D, so you could really figure out what's going on. But that's pretty much the bottom line for current source density. So you can do it 1D. If you do it 1D, then it shows you that the sources and sinks, so this is this is the sources and sinks change very rapidly with depth, indicating there's a lot of sort of vertical current flow going on. And they don't change very rapidly with as you go on to different parts of the cortex. However, if you go far enough into another cortical area, it's going to change because it's a somatosensory area, not a visual area, for instance. And so for that purpose, you can do 2D current source density on the surface of the cortex or the surface of the scalp. And that will essentially allow you to do a little bit better localization of where the currents are than just by eyeballing the evoked potential, the actual sort of voltages that you see. But in the case of an animal with a folded cortex, like a human, well, then you're still not the 2D current source density is not going to give you an accurate localization method. 3D current source density, however, would work, but it's nearly impossible to do. And so that sort of is the introduction to why we want to do source localization from scalp electrodes. Now, the problem with that is, as we'll soon see, is that you don't really quite have enough data. But there's various ways that we can sort of add in assumptions in order to give us some kind of localization method that might be capable of picking up signals that come from different cortical areas. And the cortical areas aren't too big. They're a couple centimeters across. And so what we would like to do is, we would like to know, did we stimulate the foot somatosensory cortex, or did we stimulate the frontal eye fields, or did we stimulate sort of mouth-related parts of premotor cortex, or did we stimulate, MT that, involves motion, or did we stimulate very early visual areas, or did we stimulate infratemporal cortex? So that's the kind of localization level that we would like to get out of evoked potentials, a couple centimeters. So and compared to something like functional MRI, it's a much better signal in the sense that it's the actual activity in the neurons as opposed to just the exhaust having to do with the blood oxygenation. But our spatial localization isn't anywhere near as good. And so on Friday, 8 a.m., I'll talk about the forward solution, how we sort of model how these currents get up to the scalp, and then at the 9 a.m. I'll talk about the inverse solution, giving those currents at the scalp or magnetic fields at the scalp, what are the sort of basics of trying to figure out where they came from. Okay, so any questions about current source density? No, all good. Went over, as usual, slightly. So 1D, 2D, 3D current source density. Okay, well, we'll see you guys hopefully bright and early on Friday.